Just a very quick introduction for Ben here. As William said, is an early career award recipient. Ben joined ORNL in 2018 as a staff scientist, and in 2020 was awarded that early career award for the purpose of integrating coastal wetland processes into E3SM. In addition to that early career work, Ben leads one of the main MODEX questions within the NG Arctic Phase 4 project and is also a key contributor to the Compass FME project, both in the areas of integrating wetland processes into E3SM. So Ben, please.
Yeah, thanks a lot for the introduction, and thanks very much for the opportunity to speak to this group. Yeah, I'm going to talk today about work I've been doing for about the past almost 5 years on modeling coastal wetland processes using the E3SM land model. And this work is mostly supported by an early career award from the ESMD program, but it's also benefited from a lot of collaborations with some of the other projects that Peter mentioned, including NG Arctic, Compass FME, and yeah, some various other related projects in the wetland space. And I've also benefited a lot from both some of the postdocs who worked on the project and also other collaborators at ORNL and other labs. So in general, the motivation for focusing on coastal wetlands includes several different factors that make them important both locally and also regionally and globally. In terms of their carbon cycle impact, coastal wetlands are very productive. I'm showing one way of estimating productivity, which is carbon burial rates. And coastal wetlands like salt marshes and mangroves and seagrass ecosystems can store carbon, particularly in their sediments, at very high rates of up to multiple orders of magnitude higher than ecosystems that we typically think of as being very productive, like tropical forests and temperate forests and boreal forests. This is due to a combination of having abundant water, very productive vegetation, often abundant nutrients, and also sort of ongoing burial of organic matter in waterlogged sediments. And these rates of carbon storage can be regionally and globally important, because even though in a lot of areas the sort of total spatial coverage of wetlands can be small relative to other ecosystems, they're really hotspots of biogeochemical cycling within sort of coastal regions. Aside from carbon, coastal wetlands also provide some other very valuable ecosystem services. They sit between river flows and the ocean, and they act as a filter on a lot on nutrients and other water quality related factors. So water that flows in and out of wetlands gets biogeochemically processed, and they can remove a lot of particularly nitrogen and potentially phosphorus from water before it enters larger water bodies like the ocean. Wetlands also provide some other benefits, including flood mitigation and habitat and recreational opportunities, which impact sort of a lot of different areas of society, especially in coastal regions, which are also highly populated. And so on this level, there's a lot of value in understanding what's keeping them stable and how they respond to ongoing global changes. In the context of these global changes, a lot of coastal wetlands are also vulnerable to ongoing changes. And this means that the services that they provide, including carbon sequestration, water quality, and the other ones that I mentioned, are also potentially vulnerable to loss. So I'm showing here the left two panels are from a study in coastal Texas looking at a salt marsh where hydrological flows and sea level and salinity are all changing, which is leading to some transitions from these more productive, healthy salt marsh sites towards areas where the vegetated area is dying off, patches of open water are appearing, and this type of degradation of these ecosystems, among other things, leads to carbon loss, higher methane production, and also reduces their ability to mitigate um, to mitigate impacts like storm surge from hurricanes, which then has an effect on the resilience of the urban areas and populated areas nearby. And we can also look further upstream from these more salt tolerant areas to, especially in the southeastern US, vegetated forested wetlands where there's a lot of plants that are not particularly tolerant to saltwater intrusion, which are also vulnerable to storms and other saltwater intrusion events that can lead to mortality of plants, changes in ecosystem function, and loss of services that are valuable. So in the E3SM context, we have these important ecosystems, which we would both like to be able to include in our accounting for the carbon and nutrient cycling at the continental scale, and also are useful to be able to predict how they're going to change in the future. And the model that we're using, E3SM, is designed as it's a global model. It typically has not been used to look at really high resolution variability across some of these landscapes. And so when we look at coastal gradients, it doesn't have the ability to include a lot of the really important factors that define these coastal wetlands. So in terms of connection between land and the ocean, a typical Earth system model like E3SM has a river model. It can transport water and potentially nutrients from the land into the ocean, but it's not representing all of this biogeochemical processing and productivity that happens at that interface. And so the goal of this project from the beginning was to integrate um, these highly productive coastal ecosystems into the interface between where waters and nutrients are flowing off of the land and where they can enter the ocean. And this project focused on a few different particularly important factors of representing coastal wetland ecosystems. The first of those factors is vegetation. I mentioned salt marshes and mangroves as an example of highly productive ecosystems. And more generally, on the project, we've been approaching how to define plant functional types in the E3SM land model to start including some of the really important factors that define these coastal ecosystems. So this is, I'm showing a couple of photos of salt marshes in Massachusetts and some sort of short statured mangroves in the Mississippi Delta. So we have these gradients from continental gradients from north to south. And then also a conceptual diagram of some of the key traits that we need to be able to define these coastal wetland vegetation types. Along with the traits that already exist in the way that we define plants in E3SM, so things like leaf traits, biomass allocation, plant height, which we can adjust to represent some of the distinct factors of these plant functional types. There's also processes specific to coastal wetland plants that are important to represent. And those include things like the responsive vegetation to salinity levels in the soil and in water, and also the effect of inundation on plant function. So to represent the effect of salinity on vegetation, we've introduced some additional functionality to vegetation responses to the vegetation. Basically, the ability of plants to take up water through their roots, which affects their ability to photosynthesize. And by introducing some additional plant functional type traits, we can start to represent these big differences in different species that have varying tolerances to salinity. So for example, a lot of mangrove species can tolerate really high soil salinity levels because they're specifically adapted with salt tolerance traits, such as either excluding salt from their roots when they take up water or sequestering salt in their biomass in a way that allows them to continue growing. There's salt marsh, really common salt marsh species like Spartina alterniflora and Spartina patens that are also quite salt tolerant. And then we also have salt intolerant species, which can basically stop photosynthesizing and potentially quickly die when there's saltwater intrusion. And by incorporating those functionality into land model simulations, we can then represent the impact of salinity gradients on vegetation function. So here's one example of simulating the impact of salinity on salt marsh species in Massachusetts. The bluish line here is a simulation where the vegetation is not affected by salt, and the orange line is vegetation where there is a salinity impact on photosynthesis. And basically introducing that salinity effect makes the model much more accurate in terms of modeling the productivity of this salt marsh. We're also moving sort of beyond salt marsh vegetation. We're also representing mangroves in the model. Mangroves are a very important coastal wetland biome in tropical regions. They're highly productive. They have very high below ground productivity, which introduces a lot of organic matter into the soil and also traps sediment and nutrients, and is part of the reason that mangroves sequester carbon at very high rates. And this diagram is basically showing some of the key factors that we're using to start from the existing tropical tree plant functional type that's in E3SM in the land model, and modify some key traits like allocation to leaves versus roots and leaf traits to define mangrove vegetation as a distinct vegetation type. And I want to mention that both of these are building on some modifications that we've already introduced to how plant functional types are defined in ELM through the NG Arctic project, where we've basically introduced the ability to expand the number of plant functional types in simulations across different scales, which allows us to sort of retain the existing plant functional types, but also add these coastal wetland specific plant functional types to a larger scale simulation. The second aspect of coastal wetlands that I want to talk about is biogeochemistry. I'm showing here a couple of photos from a field site in coastal Louisiana that I visited, which is part of Beth Herndon's early career award project funded through the ESS program. And she's taking a lot of measurements at these coastal wetlands, and we can see that we have a lot of vegetation growing in these waterlogged soils. And in the zoomed in part of this, you can see there's this iridescent sheen on top of the open water part of this wetland, and that is a indicator of iron cycling. That's formed from iron oxide nanoparticles that precipitate out at the air interface, and that shows that there's these sort of redox interactions of different elemental cycles interacting. That's typical of iron-rich wetlands. So the starting point for biogeochemistry is in E3SM, we have a soil organic matter cycling model, which is designed primarily to represent upland sort of oxygenated soil processes. We have different litter pools that decompose at different rates and are transformed into either carbon dioxide or um, other soil organic matter pools. And this works well in areas where you can assume that there's a lot of available oxygen to convert that decomposing carbon into carbon dioxide, which is emitted out the top of the soil. This doesn't work so well in a lot of inundated wetland systems, and particularly in coastal wetlands, there are important interactions with both iron and sulfur cycling. So in this case, we need to think about biogeochemistry as more interactions of different elemental cycles. So under inundated conditions, we can think about some solid state soil organic matter, which can be depolymerized into dissolved organic matter. In the presence of oxygen, that DOM can be decomposed into carbon dioxide. When oxygen is depleted, then there are other terminal electron acceptors that can participate in the biogeochemical microbial degradation of dissolved organic matter. So when there's iron oxides available, there are iron reducing microbes which can use iron as a substitute for oxygen. This produces more soluble iron 2 and can also decompose organic matter into carbon dioxide. Particularly in coastal ocean influenced systems, there's a lot of sulfate that's introduced by ocean water, for example, through tidal inundation, and that sulfate can be reduced to sulfide by sulfate reducing microbes, which also functions as a replacement for oxygen as a substrate for decomposition. And typically we expect that the last stage of this anoxic decomposition, which is the production of methane, really mostly occurs when these more favorable substrates have been used up. So it is important to be able to include some of these more complex biogeochemical interactions if we want to understand the carbon cycle of these wetland and particularly coastal wetland systems. The way that we're including this biogeochemistry in the E3SM land model is by actually incorporating all of those reactions into a reaction network, which we're simulating using the reactive transport model pFlowtran. And so here I'm showing this reaction network. The top of it is the same ELM solid state soil organic matter decomposition, but instead of going directly to CO2, it's connected with all of these aqueous phase reactions. So we have oxygen consuming aerobic reactions, but also fermentation and sulfate reduction, iron reduction, methane production, all of this kind of stuff. And so we've coupled that pFlowtran reaction network directly into the subsurface of ELM, and that allows us to do this more complex biogeochemistry using the machinery that's already been developed on the reactive transport side. To get into slightly more detail about this, basically we're using a previously developed interface called Alchemia, which was developed as part of the Ideas Watersheds project. And so Alchemia functions as an interface between pFlowtran and ELM being the driver model, and we're using Alchemia to solve this reaction network in each soil layer every time step within the ELM subsurface. And then on the ELM side, we're calculating vertical advection and diffusion, including both solute transport through water movement and diffusion, and also gas diffusion into the soil, which depends on factors like porosity and water content. And then it connects back to ELM for all of the other things that are calculated by ELM, which includes the vegetation model, litter inputs, plant nutrient demand, and things like that. When we apply this reaction network in a coastal wetland simulation, we can start simulating this cascade of how saline tidal inputs affect biogeochemistry. So here I'm showing averaged vertical profiles of different factors. The orange lines are a saline tidal wetland simulation, and the blue lines are a freshwater simulation. And so that saline influence, the saline water introduces a lot of sulfate compared to the freshwater simulation. The sulfate is reduced to sulfide, which consumes dissolved organic matter and converts a lot of it to dissolved CO2, which increases the dissolved inorganic carbon content. And that, through a combination of reducing the substrate available for methane production and also direct oxidation of methane, reduces the subsurface methane content. And that allows the model to simulate this very large difference between higher methane flux under freshwater conditions and very low surface methane flux under saline conditions, which is consistent with observations across coastal gradients. So an important part of simulating these coastal wetlands is being able to represent the hydrology of them, including tidal cycles. And the way that we're simulating that within ELM is by applying a hydrological boundary condition. So we're basically applying a time series of water level and salinity, which then forces the hydrology of an ELM soil column. And that allows us to simulate these time varying soil water content and salinity patterns within the ELM soil column. One example of how we're applying this going across larger scales, so going from one field site to multiple field sites, is in the Compass FME project, which is designed around these what they call synoptic sites, which are heavily instrumented gradients of wetland transition and upland plots across both a salinity gradient in the Chesapeake Bay and also a gradient of soil types in western Lake Erie in the central US. And so we have measurements across these sort of like elevation and salinity gradients. And so we've applied this model across all of those sites, and I'm showing a couple of examples here. So Portage River is one of the Lake Erie sites, so that's a freshwater site. The wetland part of it is inundated quite often. So the top line here is water level. Water table is close to the surface. It's pretty wet. Oxygen is more limited in the model. And in the transition where the water table is lower, then we have more oxygen penetrating into the subsurface. In the G Crew or the Cirque site, this is a pretty more saline Chesapeake Bay wetland. We also have this inundated wetland area versus a less wet transition area. And here we have a similar oxygen contrast, but also salinity is being introduced when the wetland is flooded, and so we have a higher salinity in the subsurface also. That's tidally driven. This then translates into some contrast in biogeochemistry, as I showed. So the oxygen is still here, but we can also see that this saline influence is also introducing sulfate into the subsurface, which propagates downward as water flows. And this leads to big differences in methane production. So again, the freshwater site is producing a lot more methane than the saline sites. And that's something that's both salinity dependent, and also we got much lower methane production out of places with more oxygen in the subsurface because the water table is lower. I want to finish up quickly by talking about how we're going upwards in scale here. I've been showing individual site level simulations, but we're also running some regional scale simulations. We're configuring those using wetland maps. So we're at this regional scale. Here we're in Southeast Texas as part of the Southeast Texas Urban Integrated Field Lab project. And so we're using the National Wetland Inventory and National Land Cover Database to map out where we have saline wetlands and freshwater marshes and freshwater forested wetlands. And by running multi-grid cell simulations with these same coastal wetland functionalities, we can start mapping out spatially variations in, for example, methane production across these sort of regional scale gradients of hydrology and salinity. And the next steps for this are expanding this up to continental scale coastal gradients. So we're working with configuring simulations at this, I believe this is a half-degree grid of coastal grid cells, and we're forcing the hydrology for individual wetland areas using this data set that Chucho Gomez Velez has been a big help with, basically using a height above nearest drainage data set combined with the national water model to calculate inundation patterns at high resolution within each of these grid cells, and then map that back out to subgrid wetland units. So to finish up with this, in summary, coastal wetlands combine a lot of different biogeochemical, vegetation, and hydrological processes, and we've been working within the E3SM land model to represent multiple factors of those. So coupling together, coupling redox biogeochemistry into the model subsurface, defining coastal wetland plant functional types, including mangrove and salt marsh and their responses to salinity and inundation, and then scaling these up to regional scales where we can start estimating regional both land atmosphere and lateral fluxes of key factors like um, carbon and nutrient cycling, and then working towards having some continental scale estimates of these key factors. And I'd like to again thank the the Department of Energy Office of Science and the ESMD program for funding this through an early career award, and also several different projects associated with the ESS program, NG Arctic, Compass FME, and the Southeast Texas Urban Integrated Field Lab, which have been great opportunities to evaluate and apply these model developments. So thanks a lot for the opportunity to talk, and I'll be happy to take any questions if we have time.